In Chapter 16, we are going to explore glycolysis and gluconeogenesis. Overall, for this chapter, we're going to look at the glycolysis pathway. We're going to explore what it is and what it does, and look at more detail into the steps involved in the process. We'll also talk about the steps where and the methods of regulation for the glycolytic pathway, and how some other precursors are going to enter the glycolytic pathway, how other things can serve as fuels. So in terms of glycolysis, here we'll look at the overview and the purpose, and then we'll go through the steps involved. And when we're exploring the steps that are part of glycolysis, you want to be thinking about what are the reactants, what are the products, what are the enzymes that are associated with that, where is the ATP being produced or even consumed, and what are the electron carrier molecules that are involved in this process. So here we're just looking at, we have this glucose, and our glucose is going to ultimately be used to break down and make ATP. That's what we want to do. And that ATP can then be used, let's say, by muscle in order for mechanical function. So glucose is broken down, and here we're in the cytoplasm, and that is where the glycolysis is going to occur. And glucose in this process will be broken down to pyruvate. And then we'll see that pyruvate has a couple of different fates. It can go on for fermentation, and here it's lactic acid fermentation, and that's under anaerobic conditions. Or under aerobic conditions, it's going to pass into the mitochondrion, where it's going to undergo further breakdown, and that's where we're going to see Krebs cycle occur. So here we're looking at this overall process of taking glucose and converting it to pyruvate, and then that pyruvate has one of three fates that we're going to go through. Fermentation, and it can be alcohol fermentation or lactic acid fermentation, and those again are under anaerobic conditions. And then under aerobic conditions, we're ultimately trying to oxidize this fuel down to carbon dioxide. So we discussed this overview in general in chapter 15, and we talked about how the ultimate goal of this was to take the fuel that we consume in the form of some kind of carbon compound and break it down to eventually produce the CO2. And throughout that process of taking fuel and getting it to CO2, we are extracting energy in the form of ATP. And we either do that directly, or we're going to do that through these high energy electron carriers that we have. So the glucose is converted to pyruvate via glycolysis, and the pyruvate then can go on for one of these other pathways. So glucose is generated from dietary carbohydrates, and we have a number of key enzymes that are going to come into play when we're trying to break down some of these carbohydrates that we consume. So these dietary carbohydrates are much more complex than these simple glucose molecules. So we actually have these alpha amylases, and we're going to see these alpha amylases in saliva, and we're also going to see them in pancreatic secretions. So these alpha amylases are going to help cleave the alpha 1,4 bonds, and it's going to help cleave those alpha 1,4 bonds of starch and glycogen. When this occurs, we're going to end up producing maltose and maltotriose. Now, we have additional enzymes that are going to help break this down further. And we see here the maltase. And maltase and alpha glucosidase, these are going to allow for the complete digestion of di and trisaccharides that are produced up here. And we're ultimately trying to form glucose from this so that we can then enter this pathway. Now, at this point, when we have this digestion of these di and trisaccharides, the result of this is going to be the production of what's referred to as limit dextrin. Okay, so this is the molecule that remains after we have our amylase digestion. This limit dextrin is going to be rich in alpha 1,6 bonds. And this alpha dextrinase can actually degrade the limit dextrin. So the role of this is to degrade the limit dextrin. Aside from that, we do have sucrose, or rather sucrase, which is going to hydrolyze sucrose. And then we have this lactase, which is going to cleave lactose. Lactose. So a number of different enzymes that are going to be key in this process of breaking down dietary carbohydrates into a form that's going to allow us to get to glucose and enter this glycolytic pathway. Now, glucose is an important fuel, and it's an important fuel for a number of reasons. It's believed that glucose was perhaps the initial fuel for cells, or it was around in these prebiotic conditions. So it may have been available for these primitive biochemical systems. We can form it under those same prebiotic conditions that allow us to form some of these simple biochemical substances, that whole primordial soup experiment. And glucose is also the most stable of the hexose sugars, and it has a low tendency to non-enzymatically glycosylate proteins. So these are kind of the reasons that glucose is the fuel, or believed to be the fuel, that we actually use rather than some other carbohydrates. So let's start to look at glycolysis and its stages. Glycolysis is thought of as occurring in two distinct stages. The whole process of glycolysis, the goal is to convert one molecule of glucose into 2 molecules of pyruvate. And with that conversion, we generate 2 molecules of ATP. But this is a two-stage process. And stage 1 is the trapping and preparation stage. So our stage 1 is trapping and preparation. Trapping and preparation. So it traps glucose in the cell and it modifies it so that it can be cleaved into a pair of phosphorylated 3-carbon compounds. And we'll see that as we move through the different reactions involved. And then, so in this stage 1, trapping and preparation, no ATP is generated here. So no generation of ATP. Now this stage 2, this is where we generate ATP. And this is going to be the harvesting stage. Because here we're harvesting the ATP or the energy from these. So stage 2 oxidizes these 3-carbon compounds to get to pyruvate. Pyruvate's going to go on to produce more ATP, but in the process of taking 3-carbon compounds and converting them to pyruvate, we're generating 2 molecules of ATP. So, and hopefully you can see this, but this is a figure in the book. This kind of shows the breakdown of these 2 stages and the reactions involved in these 2 stages. Now, do note that at the end of stage 1 here, we're actually producing 2 molecules of this glyceraldehyde 3-phosphate. So when it enters into stage 2, this process actually occurs twice. And we want to keep that in mind so that we're able to keep track of our ATP molecules. Okay, so if we're looking at stage 1 reaction 1. So glucose is going to enter the cell, and it does this through transport proteins. And once the glucose enters the cell, it's going to be phosphorylated by ATP. The phosphorylation of glucose by ATP takes place through the enzyme, or with the help of the enzyme, hexokinase. And what we form is glucose 6-phosphate. So in stage 1, reaction 1, glucose is phosphorylated by ATP via the enzyme hexokinase to produce glucose 6-phosphate. Now this is an important reaction because once this happens to glucose, it's kind of committed. It's stuck inside the cell. We're trapping it. The glucose 6-phosphate cannot pass through the membrane. And it can't pass through the membrane for a couple of reasons, one being this charge that's present on the structure, the other being that this came in via a transport protein. Well, this cannot go through that particular transport protein, and we don't have a transport protein that will move this. So the addition of that phosphoryl group, it traps it into the cell. So it traps the substance in the cell. The other thing that this phosphorylation does is it will facilitate the eventual breakdown of glucose through this process. Because it's trapped in the cell, we're going to have subsequent reactions breaking it down. And we're going to do that into molecules that possess high phosphoryl transfer potential. And creating those high phosphoryl transfer potential molecules is what will result in the formation of ATP down the road. So glucose is phosphorylated via ATP by hexokinase to produce glucose 6-phosphate, and this is where we trap it in the cell. In the next reaction, we have an isomerization, and that isomerization is taking this glucose 6-phosphate and converting it to fructose 6-phosphate. This particular process is going to be catalyzed by the enzyme phosphoglucose isomerase. Glucose isomerase. Phosphoglucose isomerase. And it actually takes a couple of steps to accomplish this overall isomerization process. And that's because if you notice, this glucose 6-phosphate is present in a ring structure. And so we have to first facilitate the opening of this ring structure. Okay, so we form this open chain form. And once we have the open chain form, then we can have the isomerization take place. So we can facilitate this isomerization, which is just this rearrangement of the substance. And once we are able to undergo this isomerization reaction, then we have to facilitate the ring closing so that we end up with this closed ring structure of fructose 6-phosphate. That fructose 6-phosphate is then going to be phosphorylated via ATP and the enzyme phosphofructokinase to produce fructose 1,6-bisphosphate. So we had the 1-phosphate here. We're going to take this ATP and transfer that phosphoryl group from ATP onto this fructose 6-phosphate to form 1,6-bisphosphate. So there's our 2 phosphate groups that are present there. So again, we have fructose 6-phosphate is phosphorylated via phosphofructokinase to produce fructose 1,6-bisphosphate. That fructose 1,6-bisphosphate, this is now going to be split into 2 3-carbon substances. We're going to cleave it into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And this is going to take place using the enzyme aldolase. Now, this glyceraldehyde 3-phosphate, we've come to the end of stage 1 here. This substance is able to directly move on to stage 2. But this dihydroxyacetone phosphate, this cannot directly move on to the next stage. First, we have to take this dihydroxyacetone phosphate and convert it to glyceraldehyde 3-phosphate. And we do that through a subsequent reaction. So here we have the dihydroxyacetone phosphate being converted to that glyceraldehyde 3-phosphate, which can then, that's our second glyceraldehyde 3-phosphate, or our second 3-carbon substance, which enters stage 2. And this process takes place using the enzyme triose phosphate isomerase. And we can look and further explore the mechanism for this triose phosphate isomerase. This figure is just showing the overall mechanism. This enzyme is significant in that it accelerates isomerization by a factor of about 10 to the 10. So it's significantly accelerated over some other types of processes. And the enzyme also has a way to prevent undesired side reactions. So what can happen with this reaction is you can have decomposition of this enediol intermediate, and that would form what's referred to as a methyl glyoxyl and orthophosphate products. But because of the catalytic mechanism of this particular enzyme, we prevent those side products or undesired products and are able to take this dihydroxyacetone phosphate and produce the glyceraldehyde 3-phosphate from that. So once we have our 2 molecules of glyceraldehyde 3-phosphate, we can enter into this stage 2, and remember this occurs twice. Now keep in mind that at the end of stage 1, we have had a cost of 2 ATP. We phosphorylated twice, so we use 2 ATP here. Okay. So up until this point, 1 molecule of glucose has been converted into 2 molecules of glyceraldehyde 3-phosphate, no ATP production, consumption of 2 ATP molecules. Now we're going to begin to get that energy harvested out. We're going to have a net ATP production in stage 2. So the first step of stage 2 involves the conversion of this glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. And this should look familiar to you because we talked about 1,3-bisphosphoglycerate in chapter 15 when we talked about substances that had high phosphoryl transfer potential. And this is a substance that has an even higher phosphoryl transfer potential than ATP. This particular reaction that takes place, so your glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate occurs through this enzyme glyceraldehyde 3-phosphate dehydrogenase. And this actually occurs as the sum of 2 individual processes. We have oxidation of an aldehyde to a carboxylic acid, and this occurs using NAD+. And then we have the joining of the carboxylic acid and the orthophosphate to form the product. And we can see this mechanism in the next slide. So here we're just looking at the mechanism that occurs in order to form this 1,3-bisphosphoglycerate. And this formation of the 1,3-bisphosphoglycerate is important because we need to create, there we are here, 1,3-bisphosphoglycerate, our substance that has this high phosphoryl transfer potential. I don't want you to worry too much about these individual mechanisms, just to understand that some of these enzymes work through multi-step processes. So we have this 1,3-bisphosphoglycerate. This is a substance that can transfer that phosphoryl group, and it does so by giving it to ADP. This is going to occur via the enzyme phosphoglycerate kinase, and we're producing 3-phosphoglycerate here. This is a substrate level phosphorylation reaction. And here we are producing ATP. We're finally getting that ATP to be generated here. So the 3-phosphoglycerate that we just produced is then going to be converted to ultimately pyruvate, but it does so through a couple of steps. So the 3-phosphoglycerate is first converted to 2-phosphoglycerate, and that occurs via the enzyme phosphoglycerate mutase. So we're just changing the position of this phosphoryl group on the backbone. It's just an intramolecular shift of a chemical group on here. That's what a mutase does. Then we have the enzyme enolase, which comes in and catalyzes the formation of this phosphoenolpyruvate. So it acts on this 2-phosphoglycerate. And this is going to be a dehydration step. So this phosphoenolpyruvate will then be converted to pyruvate. And it's the enzyme pyruvate kinase that allows that to take place. Now remember that from chapter 15, phosphoenolpyruvate is another one of these high phosphoryl transfer potential substances. So its phosphoryl group is also going to be transferred to ADP to produce ATP. So at this stage we've gone through and we have produced our pyruvate, which is ultimately the end of glycolysis. And in the process, in stage 2, we generated 2 ATP per 3-carbon molecule that came in. So per that glyceraldehyde 3-phosphate. Goodness, my pen is not working, sorry. And this occurs twice because we have 2 glyceraldehyde 3-phosphates generated in stage 1. So here in stage 2 we have 4 ATP that are generated. Remember in stage 1 we used 2 ATP, which means we have a net energy production of 2 ATP. So here's our overall reaction for glycolysis. You have glucose, you have your 2 inorganic phosphates and 2 ADP, and you have your NAD+. We're going to produce 2 pyruvate, 2 ATP, 2 NADH. 2 protons are also going to be generated in the process, and we also have water that is going to be given off throughout the process. So this is the overall reaction for glycolysis. And we can look at a summary of the reactions that we just went through. So you can see the steps and the reaction that takes place here. Just in a nice summary, we did go through all of this information. The enzyme that acts on it. We can also see and relate this back to in chapter 15 when we were talking about the different types of reactions, our reaction types here. And you can see which processes are going to be energetically favorable versus which steps would be not favorable. And so you could see where you might need to have some coupling of reactions. So overall, the process is favorable. But we do have some reactions in here that have positive values. So again, for our glycolysis, you want to be comfortable with this information. You want to know the reaction that takes place at each step and the enzyme that's acting on it. We want to also know where we have the ATP being consumed and where the ATP is being produced.